Well, good morning, or shall I say good afternoon. I am so happy to be here with you today. I say that every time, and every time I sincerely mean it. I guess a day above ground is a good day indeed. At least at this juncture it is. I can think of some pretty hard days. Nonetheless, I guess I'm thinking like this a little bit because I just came from Quest. I had a blood test, and this time of year always is filled with apprehension and concern. Several years ago, I'd gone in for a routine blood exam, and the values didn't come out right, and it looked as if I had a pretty serious type of cancer, and it was, as one could imagine, extraordinarily upsetting. And it really got me thinking quite a bit about my own life and about a lot of things. One of those things was my determination never to let anything get me down, to make sure that I proceed forward, that I look towards the future. I try to be hopeful, and whatever will be, but make the moments that I have now count. And I think that to a large extent, if ever there was a message that I could give to people that I want people to really resonate with, is that the moment is now. And so take this moment and do something with it, and don't squander our time. Time is so precious, and we have so little of it here. One of the things, I love dogs. Oh, do I love dogs? I have these 3 great dogs. I just am so blessed to have them. I just wish that dogs lived longer, because you're so involved in their lives, and they're so involved in your lives, and just seeing them get older so quickly. One of my dogs, her name's Callie. She's a black dog, and she's a mutt. She was adopted, and she's 7+ years old, I guess. And you can see that she's going gray a little bit, and she's catching up to me, and I don't like seeing that. I don't want her to catch up to me. I want her to live a long time. I love that dog. But when thinking about these things, it brings up the subject of today's conversation, which is an introduction to the mechanisms, the molecular mechanisms of cancer. Believe it or not, of course, believe it or not, everybody says this, but there really shouldn't be cancer, or certainly the rate of cancer shouldn't be what we're seeing today. It's crazy. And lots of people are saying, well, we spend so much money on trying to cure cancer. Why can't we cure it? Why can't we cure it? And well, I'm going to start to address those questions, like, why does it happen? How can it happen? What are we doing to make it happen? What can we do to make it not happen? And one time I heard somebody once say to me, I caught the cancer. And I thought to myself, what an interesting way of explaining cancer. I caught cancer, like you'd catch a cold. Well, it doesn't really work that way. You don't catch cancer. Cancer kind of develops. And that's a very important distinction between what happens with cancer and what happens with other diseases. Cancer originates from within us, and that's one of the difficulties that we have in battling it, because it is so similar that to try and differentiate cancer cells from normal cells are very difficult, because they have the same molecular underpinnings, the same DNA. And if you think about it for a moment, you probably have heard this before, but it's true that human beings and chimpanzees share approximately 98.7, approximately, and I remember 98.7 because it also happens to be what we once considered to be the average human temperature is 98.7. Turns out that that's the percentage of similarity that we have with the chimpanzees. So you say to yourself, how come chimpanzees don't look more like people, and how come people don't look more like chimpanzees? Or how come we don't act more like chimpanzees, or some Some might say, well, we haven't progressed that far. We probably do. But what it really turns out is when you think about it just for a moment, think about your own body. All right, think about all the different cells in your body, and every single cell that's in your body has the same exact DNA. You know, the same DNA that's in your eyeball is the same DNA that makes up the heart muscle, or makes up the skeletal muscle, or makes up the smooth muscle in your intestine, or makes up the epithelial the DNA within your basal epithelial cells, or whatever have you. It's all the same DNA, except of course for all the microbial that's inside of us and on top of us and so forth. The most interesting thing I ever thought about was that we have somewhere about 10 times more bacteria in our gut than we actually have somatic cells in our body. And when I say somatic cells, if you're not aware of this, whenever a scientist or a physician uses the word somatic, they're really talking about of the body. That's what the word really means, cells of the body. So that's really what we're discussing. Anyways, when we get down to it, what is it within a cell that causes it to differentiate to the point that one cell can look like a hair follicle cell and another cell develops into, a bicep muscle? What happens? And that comes down to a process of regulation. Certain genes are turned on and turned off, and based upon how those genes are turned on and turned off will determine the nature of the cell development. And the way that this happens is that in order for you to produce different protein products from the DNA within the nucleus of a cell, you have to have areas where the DNA mechanism, the DNA polymerase, has the ability to attach. And there are numerous ways that the attachment sites can be blocked or made inaccessible. And in that way, we turn off genes. And so based upon those epigenetic characteristics will in many ways determine how a particular cell develops. And that's why we have some cells that based upon the different transcription factors and the different types of methylation and acetylation and ubiquitization that occurs within the histones and so forth, making things either tighter or loosely wound, exposing genes, unexposing genes. The geometry of the DNA can all affect the way in which the expression of that DNA occurs, and that expression directly relates to the phenotype. Phenotype meaning what's being expressed. You have two concepts. One thing is your genotype. That's the collection of all the genes that exist within your DNA. And then you have your phenotypes. The phenotypes are the collection just of those genes that are being expressed. All right, so you can have certain genes within your body, but if they're never expressed, then you never have those phenotypic characteristics, or a cell doesn't have those phenotypic characteristics. So that's an important, an important distinction, to make. So as it goes into the evolution of cells, certainly we can see how if you can have cells in your body that are so varied between one that is a photoreceptor like you see within the retina of your eye, and another one is epithelial cells that you see on your skin, you can imagine that there's tremendous amount of diversity that can occur just through the way in which molecular regulation occurs. All right, because look at how differentiated all the different cells are within our body. So it's pretty cool. But it also gives us some insight as to how things can possibly go wrong. And when things go wrong, what happens? All right? Now, it turns out that our mechanism for DNA replication, it's called DNA polymerase, whenever you hear something like ACE at the end of a word, it always means that it's an enzyme. So believe it or not, the machinery that's used for copying DNA is in fact an enzyme. All right, and so this particular enzyme, DNA polymerase, is an amazing enzyme. It does fantastic things. It reproduces a DNA strand nearly perfectly. It rarely ever makes any mistakes. And when it does, on that rare occasion, like 1 in a million times, make a mistake, it has components in it that do proofreading and checking. And that proofreading and checking components revert it back so that it goes from 1 in a million to 1 in a billion. And when I mean 1 in a billion, I mean nucleotides. Nucleotides are the individual molecules that are used to designate our nuclear, our DNA, the code of our DNA, create codons and things of this sort. So this particular thing, DNA polymerase, is such a beautiful, and I do mean it is beautiful when you think about it, because not only does it replicate DNA with such a high rate of fidelity, but it does so almost in a perfect way because it does have mistakes every once in a while. And we need to have those mistakes. It's actually mandatory. If those mistakes didn't occur, there would have been no evolution. Evolution is the result of genetic mistakes that are made, changes that are made to the genetic code. And a lot of these changes happen through DNA polymerase. But the rate of mutation is so small that it doesn't adversely affect the fitness of an organism. And listen to what I said very quickly. Adversely affect the fitness of an organism. This is a very, very important concept in evolution and in biology in general. When we think of fitness, what we're really talking about is an organism's ability to survive long enough to reproduce and for that organism to have children of its own. That's what we're really talking about with fitness. So when fitness improves, it means that the species as a whole becomes better attuned to its environment and better able to survive its particular environment. And what happens is, in the course of time, traits, phenotypic characteristics that help a organism live to this point where it can reproduce gives it a significant benefit, and those characteristics remain. Characteristics that reduce fitness those genes tend to disappear because organisms that have them have less opportunity or less probability of survival and surviving to the point of being able to reproduce, and therefore those genes find themselves leaving the population gene pool. All right, that's generally how things work. But it's not like the genes pick themselves up and leave. All that happens is that greater amounts of mutations are able to occur in certain regions, essentially deactivating genes entirely. And then once the genes become inactivated entirely, it means that that area can tolerate lots of mutations occurring. And what we see is over time, these mutations occurring and then eventually turning into something that we term as junk DNA. But when I say junk DNA, and this is very, very interesting. When we look at our entire genome, our entire number of base pairs within our DNA, there's approximately 3 billion of them. Of those 3 billion, probably a little bit less than 3%, somewhere between 2 and 3%, actually are our genes. And what's really interesting is that when you look at our genes, we have, people estimate us to having anywhere between 20 and 25,000 genes. I've heard some studies say as much as 25, and I've heard many studies say 20,000. I've heard other studies say 23,000. So, this is the exact number. I still think is somewhat of a point of where I've seen varying pieces of knowledge, and I'm not able to definitively ascertain the exact number of genes there are. But what I can tell you is that if there's approximately 25,000 genes, the number of proteins that we produce is literally over 200,000. And so obviously a smaller number of genes are needed to produce a wider array of proteins, which means that at one point in time, scientists used to think that every gene was responsible for a protein. We now know that there's a mechanism called alternative splicing, which results in many different types of protein variations that can occur from one gene. So now we know that one gene has the capacity and the ability to produce many different types of protein products and other types of products that are needed within an organism itself. Anyways, I am so sorry. I have to confess something. I love talking about molecular biology and evolution, and I happen to have ADHD, and I'm dyslexic and dysgraphic. So as a result of that, I don't read from a script because I think that if I read from a script, it would sound terrible. So I need to progress this video along. So what is it about cancer that makes it different than all other cells and that actually makes it lethal? Well, the first thing about cancer that makes it lethal is uncontrolled cell proliferation. And this makes sense when you have a cell which for some reason or another now grows and grows and grows without being able to have a mechanism that stops it. You can easily see that that could create problems. In other words, it would interfere with the functioning of other tissue or other organs. And that, in essence, is what causes us to die from cancer. It's not the cancer cell itself that kills us, or even its growth. It's not that cancer is harmful per se as a particular cell, like for example, a bacteria might be. The cancer cell kills us by interfering with the homeostatic functioning of different organs and different cellular processes within our body. It in simple terms, it clogs the works up. It gets in the way. It obstructs. That's what it does. So as it grows and grows and grows, it gets in the way of more and more and more cellular and organ processes, which eventually turns into them not being able to function adequately to keep the organism alive. And that's why we ultimately die of cancer. So to answer the first question, why do we die of cancer? I would say uncontrolled cell proliferation. So that's significant. And from a molecular standpoint, think about what would need to have happen because like within a cell itself, cells just don't proliferate, willy-nilly. They just don't. And there are reasons for that because the cell itself has in it certain mechanisms that stop it from reproducing. For example, there's a p53 mechanism that essentially cuts the cell off at the very beginning during the early stages of cell reproduction. Because the first stages of cell reproduction, the very first stages, are really the stage where you go through DNA replication. And right at that stage where you go through DNA replication, there are these stop programs that can just stop it from happening. So what happens is that in cells that proliferate uncontrollably, there is a defect in that mechanism that results in uncontrolled cell proliferation. And that defect came from somewhere. Maybe the cell was exposed to a mutagen that changed the DNA in some way, shape, or form, or maybe from some previous cell replication, a mutation developed. But whatever the case is, that's one reason why what I'm trying to say is that's one area where the genome needs to take a hit in order to get cancer. The second place that our genome takes a hit is in something known as immortalization. And at first thought, you'd think, well, cell immortalization sounds pretty good to me. I wouldn't mind being immortal. I would like to live forever. I don't want to die. I want to stay young. Let my cells stay young. Cell immortalization sounds great. Not so fast. What happens is that if cells don't die, then they can, and they are also involved in cell proliferation. Oh God, I'm a stutterer too. Oh boy, I hope you're enjoying this. So when you talk about immortalization and cells can't die, and combined with uncontrolled cell growth, that is a lethal situation. Cells grow uncontrollably, and they have no shot at dying on their own. That means going through a process known as apoptosis. All right, apoptosis is programmed cell death, and and now programmed cell death sounds horrible, but it's not. It's actually what saves us from dying from cancer. All right, our cells have to be recycled, and as they get old and they become senescent, you want them to engage in the process of apoptosis, programmed cell death. Programmed cell death is really actually quite a good thing in many ways. It's actually, when we look as a molecular biologist and you look at the process itself, it's a process that requires a tremendous amount of energy. So before a cell can actually go into apoptosis, what we see is the mitochondria revving up and producing all this ADP because it's necessary. And because one of the things that you happen in cell death is that you have many organelles within a cell that contain really toxic stuff. These organelles within a cell, like the peroxisome, for example, that actually has hydrogen peroxide in it, all right, or the lysosome. All these little organelles are used for digesting unused protein products or other products that have been created and need to be recycled. And so there's this whole process of recycling that goes on in the cell. But if you were just to lyse the cell and let all these things within the cytoplasm out free, the same way that these little organelles have chemicals in it that can digest, the unused cellular proteins and other components and lipids and so forth that are produced from within the cell. They can digest the cell wall and other surrounding cells themselves. And that process of a cell death where it lyses and spreads, all these quasi-toxic types of organelles and chemicals that are within the organelles is called necroses, all right? That's cell death too, but that's not programmed cell death. And necrotic death always winds up resulting in collateral damage to other tissue, all right? So necrosis is not an optimal way for cells to die. You'd much rather than die through apoptosis, which is programmed, which means that the cell no longer, when it does die, doesn't go ahead and adversely affect, the surrounding tissue, all right? That's the whole point. And then there's the final thing, which is called Anchorage independence. Now, that's kind of a cool thing about our body. It really is. Anchorage independence is the concept that two cells need to be together, or two or more cells need to be together and communicate with one another. All the cells are saying to each other, they're sending these pro-life signals. You're good, you're healthy, you're around your friends, everything's good. Of course, that's not literally what they say, but there are messengers that are going back, these pro-life signaling messengers that are telling the cell, hey, no reason to go through, apoptosis, all right? Or rather, I should have said anoikis. Anoikis is a different process of programmed cell death too, but that happens when a cell becomes detached from its membrane. And when it becomes detached through its membrane, it goes through a programmed cell death called anoikis. And so in order for you to have metastases, a cell has to become anchorage independent, meaning it has to no longer be concerned about getting those pro-life signals anymore. It doesn't need pro-life signals to live, and therefore can go wherever it wants and attach to whatever it wants. And then if you combine that with immortalization, it doesn't die. And then you combine that with uncontrolled cell proliferation, guess what? You have a cell that's proliferating and spreading, and that's going to proliferate in an uncontrolled way, and that's going to eventually interfere with your other cellular or organ system homeostatic functions, and that's not a good thing. So that essentially is what happens, but you have to think about it this way. That got to be difficult because you need genetic mutations to occur in the area of uncontrolled cell growth. You need to have genetic mutations that allow the cell to be immortal, and you need more genetic mutations to allow the cell to be adherence independent. So those are a lot of mutations. And when you think about it, DNA polymerase only has a mutation every 1 in 1 billion nucleotides. And if you think about it, within our cell itself, our cells have about 3 billion, but those 3 billion, only 2%, 2.5% are genes. And of those 2.5%, very specific genes, like 12 specific genes, need to be altered in order for this to happen. I mean, it's almost unfathomable the amount just from a pure probability standpoint. I mean, you only get 3 at most, and you have to build up all these things over time. And how does this happen? How does this happen? And the clues to how it happens, we can see with cancers that happen more frequently. The cancers that seem to happen more frequently happen in cells which are involved in greater amounts of cell division. So the more frequently cells divide, the more apt they are to being predisposed to cancer. And so what that's telling us is that we have so much cell proliferation happening in our bodies all the time that although in any one particular cell the chance of getting cancer is extraordinarily small, when you multiply that over the number of cells that we have in our body that divide and the number of divisions that occur over a lifetime, eventually what happens is that the odds change and we do become susceptible to cancer, which is quite truthfully amazing, because one it's hard to fathom how many cell divisions must be occurring within our body over a lifetime to make cancer even remotely possible. But indeed it is. However, that's not the end of the story. It's not like, oh, there's nothing we can do about it. We are going to get cancer because, those are just the odds. Well, when we think about it, we can see that cancer emanates really in 2 ways. One, I'll say, is genetic, which means that we can inherit certain genes that make us prone to getting cancer, just like we can inherit many genes that make us prone to a wide variety of illnesses and diseases, genetically. And then there's the other way, which is acquired. Now, acquired is, I guess, what we're going to speak about just for a moment. That really involves the exposure to mutagens, things that cause mutations within our body, like, for example, radiation or the sun. So, all right, so there's been a lot of talk lately about everybody needs to get more sun. It's hurting our immune system not to get enough sun. And I don't know if that's true or not. There's studies out there. There's a lot of pseudoscience. There's a lot of people with agendas. There's a lot of papers that are being published that really don't have the true independence of what the peer review process is supposed to be. All right, the peer review process is just broken beyond belief, and it's a shame because you can't trust what you read, and there's without having personally done the data collection myself, it's very difficult to discern what's good science and what's junk science. But nonetheless, there are many the mutagen situation, and with regard to sunlight, is a real situation. Exposing your skin to excessive amounts of sun can and will result in the development of various different types of skin cancer. All right. Now, the skin cancer we're all very worried about, obviously, is melanoma. We don't want melanoma. That's terrible. Melanoma. What happens is that, typically speaking, that if a melanoma becomes greater than 1/8 of an inch in diameter, the 5-year prognosis is small. So if you see something, a growth on your body, and you're not sure, get to a dermatologist as quickly as possible. Get it removed, get it biopsied. Make sure you read the biopsy report. I've heard many situations where they go to the doctor just doesn't send it into biopsy, and all of a sudden it turns out that it's a melanoma, and if the person had just got it treated earlier on, he wouldn't have, and there are these bad stories, let me tell you, and they're real stories. Anyways, so what happens with skin cancer is that first of all, skin cancer, if it affects the top layer of your skin, has no effect. You don't get skin cancer from the top layer of your skin. The top layer of your skin is a completely differentiated cell. You get the skin cancer from the basal line, which is where the cell division occurs. And that's where you need to be concerned. And what happens in this particular case is it's not so much that DNA polymerase makes a mistake. It doesn't make a mistake. What actually happens is that the DNA gets kinked. One of the types of nucleotides there are thymine, right? And so what happens is that thymine is usually attached to its base pair. But when it's exposed to sunlight, there's enough energy to get two adjacent thymine nucleotides kind of to connect with each other, bond with each other, and that kinks up the DNA and kinks up the replication mechanism. And I'm saying this rather simply. We'll go into mechanics. I will go into all this mechanistically ad nauseum. I just haven't created the cartoons yet to do it. And so when you have this dimerization, which is what it's called, it results in the collection of mutations within a cell, and those mutations can collect over time, increasing with greater amounts of sun exposure until you get to a point where you wind up getting a melanoma, or a basal cell carcinoma, or some other type of skin cancer. Basal cell carcinoma is still something that needs to be removed and checked out, but is significantly less lethal, obviously. It's anyways, wrapping up our conversation, what I really want you to take away from this is that there's a lot of factors that go into developing cancer. Cancer is not something that's easy to acquire per se. However, there are things that we may be doing, exposing ourselves to different types of mutagens and other activities that could potentiate the development of these types of mutations within our DNA. So I guess the takeaway from this is, wouldn't it be nice for us to know what those things are that are causing us problems? Obviously, we know about smoking cigarettes, smoking tobacco. The interesting thing about that is that people have always demonized nicotine. Nicotine is not the actual drug that causes the uncontrolled proliferation or the subsequent DNA damage. It's not the nicotine itself, it's all the other stuff. But nicotine has its positive and negative effects. It's highly addictive, so negative, because we really want to try to avoid, getting addicted to things. But on the positive side, there are studies that suggest that nicotine is a tremendous antidepressant. Think about that. It works better than Prozac. Oh, Prozac. Interesting. I'm sorry that I'm going all over the place, but I just need to say this about antidepressants. Antidepressants are fine. They're good. And when I talk about antidepressants, I'm talking about the category of selective serotonin reuptake inhibitors, which is a mouthful, and I will explain what all that is in a little while. But be very wary of taking them. If you're feeling blue and you're suffering from mild depression, studies have shown that placebo effect may be just as potent as taking the serotonin reuptake inhibitor. So in other words, both have shown similar amount of efficacy. The problem that practitioners run into is that these things have been shown to be effective, and to give somebody a placebo when they think they're getting an active medication is malpractice. So it places practitioners in an awkward position, truthfully. But depression is a real thing, and having mental anguish is absolutely the worst. One of the things that I'm hoping to discuss on this channel eventually is to try to destigmatize mental health issues. It doesn't help when you have people that are truly out of their mind, committing horrible crimes, killing people, and then them getting off because they claim insanity. Certainly those types of legal decisions do very little to instill confidence in the public with regard to destigmatization of mental illness. I don't know. It doesn't seem good to me. Anyways, with all this said, I think this has been an interesting conversation. It's a start to a larger conversation about cancer, about why we haven't been able to find a cure, and why we're having such a hard time detecting it. And if you do detect it, does it mean anything? Is it something to be concerned with? Is it not something to be concerned with? Those types of things. And I think this is going to be a great conversation. I look forward to discussing this with you a little bit more. Before I go, I want to first of all take care of some business, pay the bills, so to speak. I want you to go out to Pictrus, PICTRUS.com. That's my wife's graphic photography and graphics art studio. But in particular, there's a store there, and the store has these really. First of all, it has some of my photography, which is sensational. All of my photography that. It is mine, but I've given it to Pictrus. It's available for purchase for a reasonable price. You can see it. It's on the website underneath landscapes. Those are individually printed, packaged, and sent to our clients. They are in limited supply. They're all done by hand. And if for some reason one of them gets ruined, we keep your name and we would replace a damaged photograph. However, they are limited editions, and once we get past that threshold, we don't sell anymore. So there's that, and that's out there. But even more importantly, pretty soon is going to be stuff from my son. My son is autistic, and he makes these wonderful crochet animals and other little things. And if it's not there now, it will be shortly. So keep on looking for that. And if you want, there's a really cool mug. This is, I don't know if you can see it. This is this is Mel the Hungry Puppy. I'm writing a child series called Mel the Hungry Puppy Child Series. And it's all about this yellow Labrador that because of her hunger, she got thrown out of Seeing Eye Dog School, and about all the adventures she's had subsequently. It's going to be cute with a lot of, I think you're going to like it. Well, at least your children will. I look forward to speaking with you again. I think the next conversation, I'm not sure what it's going to be on. It might be on photography. It could be on quantum mechanics. It might be on aliens. Oh yeah, I have something really cool to do. All right, if you can, tell me if you want me to do this. There's this thing going around, and it describes the genetics of a particular type of alien, a gray, I believe. A gray alien. And this paper that was allegedly written by by a biologist who was working on examining the molecular structure of the grays. He wrote a paper on this, and I want to tear it down. All right, I want to read it because first of all, this guy is definitely a biologist. There's no doubt about it. And I'm not saying that aliens don't exist. I'm not saying that at all. Or I'm not saying that there's non-human intelligence. Let me put it to you that way. I'm not saying that at all because I actually kind of believe there is. I can't say 100% because I haven't actually seen one with my own two eyes, spoken, interacted, or something like that. But I think that there's a tremendous amount of evidence that suggested that there's something. I mean, Tic Tacs, at the very least, they're there. Our government released the photographs of it. That's something. I don't know what it is, but, certainly beyond our technical capability, or at least I think it is. Maybe it's not. I don't know. Our government can keep secrets pretty good. Anyways, so I was thinking about tearing apart that paper. I think it'd be really interesting and a lot of fun. So leave comments below. Remember to like and subscribe. And it's Pictrus, PICTRUS.com. There are some calendars which are gorgeous. There's one of Westchester County. So if you're one of my friends from New York, that's where I grew up. I grew up in Westchester County. Hey, everybody. Good seeing you. Get one of these. You're going to like it. It's going to be all stuff that you're familiar with and like and appreciate. So definitely get one of those. And everybody else, I have one of Central Florida, which is where I live now. And then I also have a puppy calendar, which is, of course, of Mel the Hungry Puppy. But I'm a professional photographer. So these pictures are fantastic. It's really nice. They're lovely. They tell a story. It's really good. And basically, the company Pictrus is a company that is being developed to give autistic or adults with developmental disabilities productive work. We're trying to do something good. This isn't for our own personal gain. It's really so that we can help this community. That's what we're trying to do. So anything that you do, procuring it because they're gorgeous and they're going to be great for Christmas, and you should get it because you want it. Don't get it to support this channel. I don't want that. I would only want people to buy something that they liked and that they appreciated and that they enjoyed. Like we have this dog mug that's completely black, and when you pour in warm water, a black dog that I actually did all the artwork myself appears on the mug. It's really cool. So I mean, I want you to get that because you're going to like it. Don't get it because you think you're doing something cool by supporting this channel. You are. You're doing something really cool, but do it because you want to do it. You know what I mean? I don't want people doing things that they don't want to do. That's not cool. Anyways, you have a great rest of your day, week. I hope to get to you a little bit sooner. It does take me some time to do these because I have a terrible back problem. And like yesterday, all day long, I wanted to sit down in front of the camera and do this, and I wasn't able to. So my back, it's very bad, very, very bad. We'll talk about that more, and we'll talk about healthcare at some juncture. And I'll go through with you the ridiculousness of insurance. Oh my God, there's so much to talk about. Can't wait. Have a great rest of your day, and look forward to speaking with you soon. Bye-bye.